Ram, Jai Shri Ram, Jai Jai Shri Ram. In this video, we will discuss how nerve impulse transmit from one neuron to another neuron. Fine? Yes. We will see here how nerve impulse transmit from one neuron to another neuron. We have already discussed what is synapse. Synapse is junction between the two neurons. And if synapse is junction between the two neuron, and nerve impulse have to pass from one neuron to another neuron, that mechanism we are going to discuss in this video. Fine. Okay, I'm taking one situation where one neuron, they generated impulse. Fine. In the cell body of neuron, nerve impulse generate. Okay, fine, they generated the impulse. After generation of impulse, the nerve impulse actually reached at the end of the nerve. Fine. At the end of the neuron, it's reached. Fine. I mean, at the axon and it's reached. And end of the axon, we are calling it synaptic knob. And here we have another neuron which is receiving that impulse. Right. So the neuron which generated impulse, okay, and which bring at synapse will be called presynaptic neuron. Presynaptic neuron. So see, this is the presynaptic neuron, and the process of transfer happened at the end of this presynaptic neuron. Fine. And this is the next neuron. This neuron will be called postsynaptic neuron. The neuron which is receiving this stimulus, that will be impulse, will be called postsynaptic neuron. Right. Okay, fine. So let's see. Nerve impulse was generated, and finally the nerve impulse reached and it came to the axon end. Fine. Axon end, we all know it's lobular, and we will call it synaptic knob. Yeah, we will call it synaptic knob. Dear students, we all know and synaptic knob, lot of exoplasm present, and in that exoplasm lot of vesicles are present, and these vesicles are called synaptic vesicles. Yes, synaptic vesicles. In the synaptic vesicles, actually nearby 10,000 molecules of neurotransmitters are present. How many neurotransmitter molecule? Nearby 10,000 molecules of neurotransmitters are present. So these all neurotransmitters are stored at synaptic vesicles. Whenever presynaptic neuron bring impulse here, they bring stimulus here, they bring nerve impulse here, then in the plasma membrane of the axon of the presynaptic neuron, lot of calcium ion voltage gated channels are present. That all calcium ion voltage gated channel activate. I'm writing here, when nerve impulse reached here, the calcium ion voltage gated channel will activate, and when calcium ion voltage gated channel activate, calcium ion influx occurs through it. Rapid influx of calcium ion occurs. Fine. So when influx of calcium ion occurs, then these all synaptic vesicles which have neurotransmitter, they will start to move toward the plasma membrane, and then they will fuse in plasma membrane. Yes, they will fuse in the plasma membrane of the axon, and once they will fuse in the plasma membrane of the axon, they releases neurotransmitter. Yes, the neurotransmitter which was stored here like acetylcholine or any other neurotransmitter, that they releases into the synaptic cleft. So you may ask, sir, what is synaptic cleft? So synaptic cleft, it's a junction, it's the space present between these two neuron at synapse. We will call it synaptic cleft. Yes, we will call it synaptic cleft. Fine. This synaptic cleft is fluid filled cavity. Yes, in this the neurotransmitter like acetylcholine, dopamine, serotonin, GABA, whatever is released. Okay. Once this neurotransmitter released, this neurotransmitter have receptor on the postsynaptic neuron, because postsynaptic neuron have to receive it. So on postsynaptic neuron it have receptors. Before accepting stimulus, this postsynaptic neuron, it was positively charged from outside and negatively charged from inside. That means it was, suppose it was at minus 70 millivolt charge. Fine. It was present at the resting state. Fine. once nerve impulse, once this neurotransmitter came and they binded with their receptor, like if it is acetylcholine, so they binded with the neurotransmitter receptor like acetylcholine receptor. Fine. Once they bind it with neurotransmitter receptor, then immediately this postsynaptic neuron was changed. What happened? Lot of sodium ion influx occurs, and when lot of channel will open and lot of sodium ion will flux in, if there will be influx of lot of sodium ion, obviously this charge, in this postsynaptic neuron, from the minus 70 millivolt it come to plus 30 millivolt. That means now this postsynaptic neuron is depolarized. Yes, initially it was resting state, now its charge increased due to influx of sodium ion and it's depolarized. And this is how nerve impulse transmitted from one neuron to another neuron. I am repeating the things again. I have one neuron, innocent neuron. Yes, this neuron is so innocent. It's accepted some kind of stimulus, like one person, poked him, right? Poked on the skin only the dendrites of this neuron was present. They accepted stimulus. After accepting the stimulus, they have to transfer the stimulus to the next neuron, right? They accepted stimulus, and this is the cell body where nerve impulse was generated. Now this nerve impulse, it's flowing into the axon, and finally it's came to the axon and it's came, right? At the axon end only we have one synaptic knob, and this synaptic knob is forming junction between with the next neuron. Fine. So when the nerve impulse reach the synaptic knob, synaptic knob is lobular, and the cytoplasm of the synaptic knob, lot of synaptic vesicles are present. These all synaptic vesicles, they will start to move toward the plasma membrane when nerve impulse will reach here. Actually, when nerve impulse reach here, the calcium ion influx start from the calcium ion voltage gated channel, and once calcium ion voltage gated channel will open and calcium ion influx occurs, this all synaptic vesicle rapidly they will move toward the plasma membrane, and then they releases in a fluid filled cavity that is called synaptic cleft present between these two neurons. In synaptic cleft, neurotransmitter like acetylcholine, adrenaline, noradrenaline, dopamine, serotonin, they releases, right? And then their receptors present on the postsynaptic neuron. They will bind with their receptor present on the postsynaptic neuron like acetylcholine bind with the postsynaptic neuron receptors, and then influx of sodium ion occurs, and this influx of sodium ion will depolarize this neuron. Okay, and this is how nerve impulse transmitted from one neuron to the another neuron. Fine, students. So it is the neurotransmitter only which was transferring stimulus for one neuron to another neuron. Let's talk this neurotransmitter in detail. Actually, these neurotransmitters are two type. One is called excitatory neurotransmitter. Oh my god, excitatory neurotransmitter. And another one is called inhibitory neurotransmitter. Yes, inhibitory neurotransmitter. The excitatory neurotransmitter have glutamate, acetylcholine, adrenaline, noradrenaline. Actually, excitatory neurotransmitter are those neurotransmitter, who depolarizes postsynaptic neuron. Like acetylcholine, you see, they are depolarizing postsynaptic neuron. So they are example of excitatory neurotransmitter. Those neurotransmitter who depolarizes postsynaptic neuron, they are excitatory neurotransmitter. Fine. Example, as I told you, acetylcholine, adrenaline, noradrenaline, glutamine. We have one more type of neurotransmitter, it's so interesting. Yes, it's so interesting, it's called inhibitory neurotransmitter. Inhibitory neurotransmitter, what they do, they will hyperpolarize postsynaptic neuron. Yes, if the postsynaptic neuron is depolarized, they will hyperpolarize it. Yes, and when they will hyperpolarize it means the postsynaptic neuron is inhibited. That's why we are calling it inhibitory neurotransmitter. Gamma aminobutyric acid is chief type of inhibitory neurotransmitter present in brain. Then we have serotonin, and then we have dopamine, and many more glycine we have. Now we will see how this gamma aminobutyric acid work. Actually, when the gamma aminobutyric acid will also release in the same way. Presynaptic neuron brings stimulus here, right? And the vesicles which have gamma aminobutyric acid, they fuse they fuse in the plasma membrane, and then they releases gamma aminobutyric acid. This gamma aminobutyric acid bind with the receptor present in the postsynaptic neuron, and then they causes influx of chloride ion. Now student, tell me, if chloride ion influx occurs, it's going to increase the positive charge or decrease the positive charge? Obviously it will decrease the positive charge, and when they will decrease the positive charge, that means postsynaptic neuron is hyperpolarized, my students. Yeah, so this gamma aminobutyric acid GABA causes influx of chloride ion, and this influx of chloride ion actually they hyperpolarizes it postsynaptic neuron. Fine. Is it clear, students? So this is what difference between excitatory neurotransmitter and inhibitory neurotransmitter. Fine. Fine, students. Okay, fine. So this is excitatory neurotransmitter and inhibitory neurotransmitter. Okay. Now after releasing, students, okay, fine, everything is okay, neurotransmitter release, they binded, they causes excitation, they causes inhibitants. After that, what will be fate of that neurotransmitter? Like if acetylcholine came and it's binded here, after that what is fate of that neurotransmitter? Actually, some of the extra neurotransmitter, is absorbed by astrocyte cells present here. We have glial cells. Yes, the astrocyte is a major type of glial cells which are present in this region. They will absorb extra neurotransmitter. This is one this is how this is one way this my neurotransmitter eliminate from there. Second, for specifically for different type of neurotransmitter, different type of enzymes are present. As for example, I am taking one example. In postsynaptic neuron, one enzyme present, that is called acetylcholine esterase. Yeah, acetylcholine esterase present. This acetylcholine esterase, what do you know? This acetylcholine, once the acetylcholine will bind, they will break this acetylcholine esterase will break this acetylcholine into acetic acid and choline, and they will degrade them. This is second way, right? Some enzyme release and they broken it, right, and they eliminated the neurotransmitter. There is one more way. This presynaptic neuron only releases neurotransmitter, again presynaptic neuron will reuptake it. Yes, as for example dopamine issue. This dopamine, how it's work, yeah, for this dopamine hormone, for its releases in a pleasure feeling, it releases. Like we have one presynaptic neuron, it released dopamine synaptic cleft, it binded with the dopamine receptor in postsynaptic neuron and showed the action. After that, this dopamine, they will return and they will go and they will bind with dopamine transporter in presynaptic neuron, and this dopamine transporter will reuptake this dopamine. Fine. Dopamine released, binded with dopamine receptor on the postsynaptic neuron, show the action, again they reuptaken by dopamine transporter in the presynaptic neuron. Have you heard cocaine? Cocaine? Cocaine, nice thing, right? Drug addiction, yes, very dangerous, never take it. This cocaine, what you do, when the person neuron releases dopamine, this cocaine block the reuptake of the dopamine. Fine. How? They will block the dopamine transporter. It was the dopamine transporter only, they were reuptaking it back. So cocaine, cocaine will block that. That's why person, he'll feel lot of pleasure, but that pleasure is not true pleasure, that is drug addiction. Actually, we are calling that euphoria. So in this video we discussed how nerve impulse transmit from one neuron to the another neuron, and then we discuss what are the types of neuron, and then we discuss that what will be the fate of neuron once they will bind with the postsynaptic neuron. Remaining we'll see in next video.